All right, now that we're done differentiating the pyramidal and extrapyramidal system, let's proceed with the basal ganglia. The basal ganglia is one of the many structures that help in modulating our movement. So, the basal ganglia, or the more correct term, basal nuclei, because if you remember with our anatomical terms, when we use the word nuclei, it should pertain to a collection of cell bodies found within the central nervous system. But when we use the term ganglion, this is the term for a collection of cell bodies found within the peripheral nervous system. And the basal ganglia is actually found within the brain. So it's a part of the central nervous system, not the peripheral nervous system. So it's a misnomer. So let's start using basal nuclei instead of ganglia. It's been a tradition. A lot of references use basal ganglia, but again, the correct term would be basal nuclei. So the function of your basal nuclei is for, just like your extrapyramidal tracts, is for posture and controlling or modulation of voluntary movement. And I want you to remember that this is for voluntary movement because oftentimes a lot of people get confused that the basal nuclei is for involuntary movement. So, no, it's also for voluntary movement. Specifically, it modulates the amount of movement that is needed to be produced. So, it's a voluntary function. Okay? The basal nuclei also has no direct connection with the spinal cord. So that's fascinating to know. And it's kind of makes you question how the basal nuclei is able to communicate with our body without having a direct linkage between the spinal cord, because that's our bridge to our muscles in the peripheral system, right? So we'll discuss how basal ganglia is still able to communicate its information pertaining to modulation, the correct intensity of movement needed to produce an action to the spinal cord without having a pathway directly to our muscles. So again, the basal ganglia, these are collection of cell bodies that are scattered, embedded inside our brain. And we have different names for the parts that comprise the basal nuclei. We have first the neurologic structures. We have three parts: the corpus striatum, the amygdaloid nucleus, and the claustrum. These are just mentioned as a name to identify the neurologic structures of the basal nuclei, but the more important parts would be the actual nucleus that are found or that makes up your basal nuclei. And these are the caudate nucleus, the globus pallidus, the putamen, the claustrum, the caudate nucleus plus lentiform nucleus or the lentiform nucleus, and we have the amygdaloid nucleus. So these are the more important structures that we need to discuss in this topic. Now, let's discuss the five subcortical nuclei that comprise the basal nuclei. We have the caudate nucleus, the putamen, and the globus pallidus. These three nuclei makes up your basal nuclei. However, the subthalamic nucleus and the substantia nigra, these are not exactly part of the basal nuclei. So they don't make up the structure of the basal nuclei. Instead, they play a role that is important for the function of the basal nuclei in general. Again, they are not directly part of the basal nuclei, but they play a role in the function of the basal nuclei. So the corpus striatum, it's called striatum because of its appearance. It has striations. It's striated because of the strands of gray matter that are passing through it in connecting the caudate nucleus to the putamen of the lentiform nucleus. So the corpus striatum is basically the connection between your caudate nucleus and the putamen. In this picture, you can see the striations of the corpus striatum. So that color green structure, that is the corpus striatum that connects again the caudate nucleus and the putamen. And this is such an important structure because the um, information or the input that will be coming towards the basal ganglia or the basal nuclei will pass through the structure, the corpus striatum. So, since a lot of afferent fibers passes inside the basal nuclei through the corpus striatum, this is the major site of input to the basal nuclei. It receives afferents via the corticostriate projections from the cerebral cortex, especially the sensory motor cortex, which are your Brodmann areas 4 and 312. Aside from that, it also receives afferent input from the premotor cortex and the frontal eye field. It is excitatory. When we say excitatory, it stimulates the basal nuclei. It also receives input from the intralaminal thalamic nuclei, substantia nigra, amygdala, hippocampus, and midbrain raphe nuclei. So the corpus striatum, it's like a bridge between the nuclei found within the basal nuclei and other parts of the brain, such as the thalamus. And again, I want you to remember that the corpus striatum is excitatory. So it stimulates the basal nuclei or the structures that it connects to the basal nuclei, such as the putamen. Next is the amygdaloid nucleus. This is situated in the temporal lobe close to the uncus, considered to be part of the limbic system. It can influence the body's response to environmental change, and in the sense of fear, for example, it can change the heart rate, blood pressure, skin color, and rate of respiration. If you recall, in our limbic system, it is it's part of our nervous system that is responsible for the sense of threat, the survival instinct in us is provided by our limbic system. And part of the limbic system as well is the amygdaloid nucleus. It is close to the location of the limbic system and even the hippocampus, which is found underneath the temporal lobe. And that's why it has a connection with our response to fear. So not only does it function for that survival instinct, but also for the modulation of the appropriate movement or response needed in that situation. Next, the claustrum. This is a thin sheet of gray matter that is separated from the lateral surface of the lentiform nucleus by the external capsule. The function of the claustrum is still unknown. So it's a nice to know information. At least we are familiar that this is a part of the neurologic structure of the basal nuclei. And then we have the two nuclei, the substantia nigra and the subthalamic nuclei. These are closely related to the basal nuclei because of its function. Again, it's not directly part of the basal nuclei. Instead, it works with basal nuclei. So the neurons of the substantia nigra are dopaminergic and inhibitory and have many connections to the corpus striatum. But we'll be more specific later in the motor pathway because the dopamine can also act as excitatory depending on the receptors that it will bind to. But majority of the dopamine neurotransmitter is inhibitory. Next is the neurons of the subthalamic nuclei. These are glutaminergic, which means it's excitatory, so they're very opposite in function. Again, the substantia nigra is mostly inhibitory, while the subthalamic nuclei are excitatory and have many connections to the globus pallidus and the substantia nigra. Again, I want you to remember the neurotransmitters that are being used for either excitatory or inhibitory function. So if it's for inhibition, the neurotransmitter responsible for that would be dopamine and your GABA. We'll talk about that later as well in our motor pathway. And then for excitatory function, the neurotransmitter that we use for that is glutamine or the glutaminergic neurotransmitters. So again, here are the terminology commonly used to describe the basal nuclei. We have the neurologic structures, the caudate nucleus, the lentiform nucleus, the claustrum, corpus striatum, and the amygdaloid body. So the function again of the claustrum is still unknown, but the corpus striatum is basically the connection between the caudate nucleus and the putamen. The caudate nucleus and the putamen, these are the actual nucleus of the basal nuclei. And the amygdaloid body contains the amygdaloid nucleus, which is important for the responses for whenever we are under threat. So first of the basal nuclei is the caudate nucleus. So this is a large C-shaped mass of gray matter that is closely related to the lateral ventricle and lies lateral to the thalamus. So the bulge part there of the basal nuclei is actually the caudate nucleus. And in Huntington's disease, we'll talk about that in our laboratory session, this is actually lost. So the C-shaped there is the caudate nucleus. The caudate nucleus works in junction with the putamen. That's why the corpus striatum connects these two together. That's also the reason why they work together. So the caudate nucleus and the putamen, they are both inhibitory to the globus pallidus. Again, we'll discuss this again in the motor pathway. Next is the lentiform nucleus. So this is a wedge-shaped mass of gray matter that is found like the head of the caudate nucleus. So this area is the lentiform nucleus. I have another picture here. The blue area, the blue part is the lentiform nucleus. The green one is the C-shaped caudate nucleus. And then we have the amygdaloid body. It's like found in the tail of the caudate nucleus. And then the important information that you need to remember here are more about the anatomical locations of the different nuclei. Okay, so the lentiform nucleus is found more anterior and it is medial to the putamen. The caudate nucleus, this one is the C-shaped structure and it is more medial to the lentiform nucleus. Or another way to describe its location is that it is close to the thalamus. So the caudate and the putamen, these are very strong inhibitory nuclei that inhibits the globus pallidus internal. And they use the neurotransmitter GABA to do that function. Okay, so I have mentioned earlier the different neurotransmitters, and I've said that dopamine is also an inhibitory neurotransmitter. That is correct, but that is being used not by the caudate and the putamen, but rather it is being used as a neurotransmitter of the substantia nigra. Again, dopamine comes from the substantia nigra, and it's mainly inhibitory, but there's also an excitatory function of the dopamine depending on the receptor that it binds to. For the caudate and the putamen, these are the direct nucleus of the basal nuclei, and they are inhibitory. They use GABA as a neurotransmitter and not dopamine. Next is the globus pallidus. We have first the internal. This is the first major outflow nucleus of the basal ganglia. It sends inhibitory axons to the ventral nuclei of the thalamus. What I want you to remember is the basal nuclei communicates with the thalamus via the globus pallidus. Okay, and the globus pallidus, specifically internal, is always inhibitory to thalamus. The axons from the globus pallidus projects to the thalamus, specifically again to the ventral nuclei, or the ventral anterior and ventral lateral nucleus of the thalamus. Okay, do not confuse this with the third order neuron of the somatosensory system. because in the somatosensory system, the nucleus responsible for receiving sensory input is the ventral posterolateral nucleus and the ventral posteromedial nucleus for the face and the trunk. Okay, so different nucleus in the thalamus is for the motor system. Again, that is the ventral anterior and the ventral lateral nucleus of the thalamus. After that, it travel in small bundles. Specifically the name is ansa lenticularis and the lenticular fasciculus, also known as the H2 field of Forel, before entering the thalamus. Again, the axons of the globus pallidus internal that projects to the thalamus will form these pathway or like a bundle of fibers called the ansa lenticularis and the lenticular fasciculus. We have a picture later to show you that structure. So here is the ansa lenticularis and the lenticular fasciculus. As you can see in the picture, it's just a pathway of the collection of bundles of axons coming from the globus pallidus going to the thalamus. So it's again named as ansa lenticularis, this one. And then we have you can't see it here, but we also have the lenticular fasciculus, which is another structure that is the collection of the projection of axons of the globus pallidus internal to the thalamus. So here's another picture showing us a different nucleus in the basal nuclei. So now let's talk about the feedback loop. Remember I told you earlier that the basal nuclei has no direct connection to the spinal cord. So how does it modulate movement if there's no connection between the two? And that's because of the feedback loop. So it's called a loop because it starts from the cortex and ends back at the cortex. So from the cortex, it will send afferent input to the striatum. And remember the striatum, this is the collection of the caudate nucleus and the putamen. It's the one that binds them together. So collectively, they are called the striatum. And the function of the striatum is to inhibit the globus pallidus internal. So the globus pallidus internal is the one that has communication between the thalamus. The thalamus will serve as like the secretary that will send the message from the basal nuclei to the cortex. And the cortex is the one or is the bridge that connects the basal nuclei to the spinal cord. So I hope that you understand the feedback loop. Again, from the cortex, it will go to the basal nuclei. The globus pallidus internal specifically is the one that will communicate with the thalamus. The thalamus is now a part of the brain, so there's a connection between the basal nuclei and the brain or the cortex through the thalamus. From there, the cortex will now send the appropriate response or movement based on the message of the basal nuclei to the spinal cord. So there's an indirect relationship between the basal nuclei and the spinal cord through the primary motor cortex. But we also have to identify that there's two feedback loops in the basal ganglia pathway. We have the direct pathway and the indirect pathway. So the one that I have just described is the direct pathway. It's called direct because it is directly using the nuclei of the basal nucleus. The indirect will need the help of the other nucleus that I've mentioned earlier are not part of the basal nuclei structure, but plays a role in the function of the basal nuclei. And those are the subthalamic nuclei and the substantia nigra. So those are the two structures forming now the indirect pathway. And because I've mentioned it, let's talk about substantia nigra. So this is the second major outflow nucleus of the basal nuclei. The first one again is the globus pallidus internal. This is reciprocally connected with the putamen and the caudate nucleus, and this one specifically release dopamine as a neurotransmitter to inhibit the striatum. Again, I've mentioned dopamine can also be excitatory depending on the receptor that it binds to. So If it binds to D2 receptors or D2 dopamine receptors, that's for inhibitory dopamine. If it binds to D1 dopamine receptors, that is when it becomes an excitatory neurotransmitter. And this is specifically being released in the substantia nigra called pars compacta. And the fibers, or this will be projected towards the striatum, which is again inhibitory to the globus pallidus internal. So the substantia nigra will inhibit the function of the striatum. And remember, the function of the striatum is to inhibit the globus pallidus internal. So if there's no inhibition coming from the striatum, the globus pallidus internal will be able to do its function, which is to inhibit the thalamus. So I hope that you are not getting confused with the terms inhibit, because I'm using it a lot, but that's what mainly the function of the basal nuclei is. Unfortunately, it's more of inhibition of the function of another inhibitor. So again, the substantia nigra, this one is the second major outflow, specifically for the indirect pathway. Later we'll discuss that under the motor pathway. So we have mentioned the pars compacta as a part of the substantia nigra that releases dopamine, but we also have the pars reticulata of the substantia nigra that receives input from the striatum. It sends axon outside the basal ganglia to modulate head and eye movement. So again, the pars compacta is the one that releases dopamine. The pars reticulata is the one that receives. So the one way to remember this is that it has the word re in its name. So it receives input from the striatum. Next is the subthalamic nucleus. This is also known as the nucleus of Lewis. It receives inhibitory input from the globus pallidus and the cortex. But its function is also to inhibit. Specifically, it sends an inhibitory response to the globus pallidus. So it receives inhibitory input from the globus pallidus, and it also sends inhibitory input to the globus pallidus. It's an important relay and modifying station, specifically for the indirect pathway. So before I proceed with the pathway, let's just talk about more of the anatomical structures of the basal nuclei. So here is a picture of an MRI showing us the different basal nuclei. We have the caudate nucleus, the anterior commissure, the putamen, insula, the globus pallidus, thalamus, optic radiation, and the optic lobe. And again, the function of the putamen and the caudate nucleus is inhibition. It's an inhibitory nucleus to the globus pallidus. The globus pallidus is the one that connects to the thalamus to inhibit it. The thalamus will then send that information to the cortex. Let's also talk about the internal capsule. So the internal capsule contains critically important pathways such as the corticobulbar and the corticospinal tract. So remember, your corticospinal and corticobulbar, they are part of your pyramidal system. So it means that the pyramidal system passes through the internal capsule. So when you have a damage or lesion to the internal capsule, it means that motor problems will occur because the axons that passes through this structure are mainly motor fibers. Now the anterior capsule can be divided into two parts. You have the anterior limb and the posterior limb. The anterior limb separates the lentiform nucleus from the caudate nucleus, and in a way it resembles the shape of the caudate nucleus, right? It contains the thalamocortical and corticothalamic fibers that join the lateral thalamic nucleus and the frontal lobe cortex. Basically, it contains fibers, descending fibers that are important for movement, not just of the body, but also of the face, specifically the thalamocortical, the corticothalamic fibers. Next is the posterior limb. This is located between the thalamus and the lentiform nucleus. It contains the major ascending and descending pathways. The corticobulbar and the corticospinal tract is found within the posterior limb, with the fibers to the face and arms in front of the fibers to the leg. So, the clinical importance of these parts is just knowing that if the posterior limb is the one damaged in the internal capsule, it could suggest that there would be problems with the major pathways, specifically of the corticospinal and the corticobulbar tracts, because we know anatomically this is where they are located. Whereas if it was in the anterior limb, it's mainly affecting the thalamocortical fibers or the corticothalamic fibers, which are responsible for the afferent and the ascending and descending tracts to the face. Now let's watch this video of the basal ganglia. This is again for visual learners. I think it's more interesting learning with pictures because you can identify the structures and you can easily see and understand the anatomy and the locations of the
The
structures.
basal
So let's watch
ganglia
this
or basal
video
nuclei is a
before we
structure located
discuss
deep within the
the
brain,
last part
and it's made up
the
of a group
motor
of
system,
nuclei,
which is the motor
so
pathways
millions of
under
nerve cell
basal
bodies.
ganglia.
Put simply, the cerebral cortex decides how it wants to move the body and sends that input to the basal ganglia. And then the basal ganglia's job is to help execute a smooth movement. The basal ganglia are actually two pairs of deep structures, one on the left side and one on the right side of the brain. Each pair consists of the globus pallidus, which has the internal globus pallidus and the external globus pallidus, as well as the striatum, which includes the caudate nucleus and the putamen. The basal ganglia is linked to other brain structures, like the ventral anterior nuclei and the ventral lateral nuclei of the thalamus, as well as the substantia nigra of the midbrain. The basal ganglia can help start, stop, and control desired movements, while also inhibiting undesired movements. As an example, when you walk, you have to move one leg at a time. So the basal ganglia helps one leg to step forward, while inhibiting the other leg, so that it's stationary, and that prevents you from falling. Additionally, the basal ganglia is involved in perception. Let's take a look at this picture as an example. You can either see a rabbit with its two long ears, or a duck with its beak. And you can choose which animal to see, but you can't see both simultaneously, because the basal ganglia stimulates the vision of one while it inhibits.
So now let's talk about the motor pathways. We have two motor pathways, the direct and the indirect pathway. These pathways are neural inputs into the basal ganglia that comes from the cortex, so from different areas of the brain, such as the parietal, frontal, and the motor areas of our brain. These inputs coming from the brain or the cortex are excitatory. They are always excitatory, and they use glutamine in order to produce that stimulus. So they enter the basal nuclei through the striatum. Remember, I told you that one reason it's called striatum is because of its appearance, wherein a lot of axons passes through that structure. That's why it causes a striated appearance. And one of the axons that passes through there is axons coming from the cortex. So again, the striatum is like the gate, the passageway between the cortex to enter the basal nuclei, to excite the basal nuclei. So these inputs are then relayed by a number of different circuits throughout the basal nuclei to produce a final output from the internal globus pallidus to the thalamus. So it will send like a question to the basal nuclei. The brain will send an information or an afferent input to the basal nuclei. It will ask what is the appropriate response, or what muscle should be inhibited, what muscle should be excited to the basal nuclei. And then the striatum, which receives that information, will now send that to the basal nuclei from different connections, from different circuitries, and that is the direct and the indirect pathway. The major outflow will be coming from, again, the internal globus pallidus. And the second major outflow will be coming from the substantia nigra. The internal globus pallidus produces a tonic inhibitory output to the thalamus, which is then relayed as an inhibitory signal to the motor cortex. So it means that if the internal globus pallidus sends a stimulus to the thalamus, it will inhibit it. It will stop the thalamus to send signal to the cortex, and that will be interpreted as inhibition. It means that, okay, this muscle is not supposed to move. This one should be inhibited. So again, the internal globus pallidus is the one that will inhibit the thalamus. If the thalamus is inhibited, or if there's a tonic inhibitory output to the thalamus, this will be interpreted as an inhibitory signal to the motor cortex. Now, the direct pathway, this involves excitatory stimulation from the cortex and the substantia nigra to the striatum. This excitation of the striatum produces an inhibition of the internal globus pallidus. As I've mentioned earlier, the cortex in general is excitatory, and it will excite the striatum because it's the only connection they have to the basal nuclei. When the striatum is excited, its function is to inhibit the globus pallidus internal. So if the cortex stimulates the striatum, it will then inhibit the globus pallidus internal. If the globus pallidus internal is inhibited, it cannot do its function to inhibit the thalamus. Therefore, it will be interpreted as an excitatory output to the cortex. So overall, the direct pathway is an excitatory output to the cortex. It will produce a movement, it will produce a contraction, because the internal globus pallidus was inhibited by the striatum. That is again under the direct pathway. In the indirect pathway, this is the opposite of the direct because it causes an inhibitory output to the cortex. It utilizes the direct pathway with two additional intermediate steps. Again, excitatory input coming from the cortex to excite the striatum. The striatum will then inhibit, this time, the external globus pallidus. The inhibition of the globus pallidus in external leads to less inhibition of the subthalamic nucleus. The subthalamic nucleus is an excitatory input to the internal globus pallidus. So if there's less inhibition to the subthalamic nucleus, it means that it can definitely excite the globus pallidus internal. And what is the function again of your globus pallidus internal? It is to inhibit the thalamus. And so, if the globus pallidus was successful in inhibiting the thalamus, this will be relayed as an inhibitory output to the cortex. And that is how the indirect pathway is an inhibitory output to the motor cortex. So once again, we have two pathways, the direct and the indirect pathway. The direct pathway will produce an excitatory output to the motor cortex. The indirect pathway will produce an inhibitory output to the motor cortex. Let's start again with the direct pathway. So this one will start from the cortex. It will send an excitatory input to the striatum. The function of the striatum is to inhibit the globus pallidus internal. So if they are excited, they will then be able to inhibit the globus pallidus internal. Now, the function of the globus pallidus internal is to inhibit the thalamus. If the striatum inhibits the function of the globus pallidus internal, it will not be able to inhibit the thalamus. The thalamus will therefore be able to send an excitatory output to the motor cortex. Next would be the indirect. So the indirect, this one will not be coming from the primary motor cortex. Instead, it will be coming from the substantia nigra. However, the substantia nigra is also located within the brain, so in a way, it's still coming from the cortex. But in the indirect, this one will be using dopamine as a neurotransmitter. So the substantia nigra, specifically at the pars compacta, it will release dopamine. And again, dopamine is either inhibitory or excitatory, but for this one, it will excite the striatum. Once the substantia nigra excites the striatum, it will therefore inhibit the globus pallidus external. And this is the reason why it's indirect, because it's not directly inhibiting the globus pallidus internal. So once the globus pallidus external is inhibited, it cannot therefore do its function to inhibit the subthalamic nucleus. And the subthalamic nucleus is like a partner in crime of the globus pallidus internal. It stimulates the globus pallidus internal. So if the globus pallidus external cannot inhibit the subthalamic nucleus, it can then stimulate, the subthalamic nucleus can then do its job to stimulate even further the globus pallidus internal. And so the globus pallidus internal will be able to inhibit the thalamus, and there will be no input to the motor cortex, and this will be relayed as an inhibitory output, or no contraction or no movement. And that is again the two pathways of the circuitry happening in the basal nuclei. So in summary, these are the functions of the different structures that makes up the pathway of the basal ganglia circuitry. So the striatum again, this one will inhibit the globus pallidus, both internal and external, depending on the pathway. If this is in the direct pathway, it will inhibit the globus pallidus internal directly. If it's in the indirect, it is coming from the substantia nigra, and then it will inhibit the globus pallidus external. The substantia nigra, its function is either inhibitory or excitatory, but it will excite the striatum in the indirect pathway. The globus pallidus internal inhibits the thalamus. The globus pallidus external will inhibit the subthalamic nucleus, and the subthalamic nucleus will excite the globus pallidus internal. Again, they're like partner in crime. And then lastly, the thalamus. The thalamus is the connection of the basal nuclei, efferent or outflow or output to the cortex. So if there's stimulation of the thalamus, it will be relayed as excitatory output. If there's inhibition of the thalamus, it will be relayed as inhibitory output to the motor cortex. So it's like it's the sender of message from the basal nuclei to the cortex.
For the basal ganglia to work, nearly the entire cerebral cortex projects onto the striatum. The striatum then projects onto the thalamus, and from there neurons head back to the cerebral cortex through two pathways: the direct pathway, which is excitatory, and the indirect pathway, which is inhibitory. So the direct pathway and indirect pathway have to be carefully balanced to control smooth movement. Now, there are two main neurotransmitters involved in these pathways: the excitatory neurotransmitter glutamate and the inhibitory neurotransmitter GABA. In the direct pathway, the cerebral cortex sends excitatory projections to the striatum. Then the striatum sends inhibitory projections to the internal globus pallidus. Then the internal globus pallidus sends inhibitory projections to the thalamus, which is usually in an active state. With this setup, if the striatum inhibits the internal globus pallidus, then the internal globus pallidus cannot inhibit the thalamus. The two negatives cancel out. As a result, the thalamus is free to send excitatory projections to the motor cortex, and this initiates voluntary movements. In the indirect pathway, the cerebral cortex sends excitatory projections to the striatum once again. But this time the striatum sends inhibitory projections to the external globus pallidus, rather than the internal globus pallidus. Then the external globus pallidus sends inhibitory projections to the subthalamic nucleus, which are constantly active. Similar to before, the result is that if the striatum inhibits the external globus pallidus, then the external globus pallidus cannot inhibit the subthalamic nucleus. Once more, the two negatives cancel out. And as a result, the subthalamic nucleus is free to send excitatory projections out. It just so happens that the subthalamic nucleus sends its excitatory projections to the internal globus pallidus. This is in direct opposition to what happens in the direct pathway. By exciting the internal globus pallidus, it's able to inhibit the thalamus, which sends excitatory signals to the motor cortex. But the striatum also receives input from another brain structure, the substantia nigra, which releases a third neurotransmitter called dopamine into the striatum. Dopamine acts on both the direct and the indirect pathways, but with opposite effects due to the presence of two different receptors: D1 receptors, which are excitatory, and D2 receptors, which are inhibitory. D1 receptors are expressed by the striatal cells that project to the internal globus pallidus. When dopamine is released by the substantia nigra, it binds to D1 receptors, and that activates the direct pathway that controls the muscle you want to move. On the other hand, D2 receptors are expressed by the striatal cells that project to the external globus pallidus. When dopamine binds D2 receptors, they inhibit the indirect pathway for that same muscle. Ultimately, the action of dopamine on the striatum favors the excitatory projections to the motor cortex to reinforce the desired movements. In other words, if you were to add abnormally high levels of dopamine, the direct pathway for wanted movements becomes more active. And at the same time, the indirect pathway for inhibiting unwanted movements becomes less active. As an example, individuals using cocaine or ecstasy often have enhancements of movements they want, like dancing at a rave. But they're less able to control unwanted movements like grinding their teeth. Alright, as a quick recap. The basal ganglia is made up of the striatum and the globus pallidus. The striatum receives input from the cerebral cortex about a desired movement, and then it sends output to the other basal ganglia structures through the direct and the indirect pathways. The direct pathway ultimately leads to activation of the thalamus, which then stimulates the motor cortex to initiate wanted movements. On the other hand, the indirect pathway ultimately leads to inhibition of the thalamus, to inhibit undesired movements.
For the last part of our motor system, we'll also discuss the cerebellum. So the cerebellum, this is also known as the small brain because it looks like a brain also just underneath the cerebral cortex. So the cerebellum, this also plays an important role in movement because it can process and interpret information from the motor cortex and also receive information from sensory input or from sensory system that specifically is for posture and balance. So it coordinates those information together, and from that it is able to produce a smooth coordinated activity, which is very important if we want to do a skill, for example. So cerebellar processing fine-tunes motor activity by ways of receiving or processing or interpreting information from the motor areas via relay nuclei in the brainstem. Then from there, it will notify the cerebellum to either initiate a voluntary muscle contraction, and then the sensory system will come in place through the proprioceptors throughout our body regarding the tension in the muscles and tendons and joint position. So because of that information from the sensory system, it can now have an idea about our body in space. So it will relate the information coming from the motor area and the information coming from the sensory system. So the cerebellar cortex will then calculate the best way to coordinate all of the important components such as the force, direction, and extent of muscle contraction to prevent incoordinated or overshooting, for example, when you're trying to reach for your phone. It's the function of the cerebellum to make sure that it is only within enough distance by gauging or by having an idea of the visual spatial location of the phone and by knowing how much length is needed by the muscle to reach for the phone, that is being calculated by the cerebellum. If there's no cerebellum, the chances of overshooting and not being able to reach exactly where your phone is could occur. And aside from that, it will also maintain posture, ensure smooth coordinated movements. Then we have the peduncles found within the cerebellum. These are connections within other areas of the brain, such as the motor cortex. So, through this structure, the axons or the nerve fibers coming from the cerebellum is able to project its input towards the cortex. And we'll discuss that later. So, the cerebellum, there are portions of the thalamus project by way of the central tegmental tract to the inferior olivary nucleus. This nucleus in turn sends fibers to the contralateral lateral cerebellar cortex. So the olivary nucleus, this one, if you look at it anatomically, it's very close to the cerebellum, and that's why they share connection. And this is how the thalamus is able to communicate with the cerebellum. Again, it's through the olivary, specifically the inferior olivary nucleus, that it can communicate with the cerebellum. From the cerebellum, the loop to the thalamus is closed by the dentate and the contralateral red nucleus. So the cerebellum will end its communication to the centers or nucleus found in the brainstem, such as the red nuclei. Projections from the globus pallidus of the basal nuclei to the red nucleus converge with inputs from the motor cortex and the deep cerebellar nuclei. So that is how the cerebellum is a part of the extrapyramidal system because of its connections with the basal nuclei and the extrapyramidal tracts, such as the red nucleus and other centers in the brainstem. So the two major functions again of the cerebellum is for coordination of voluntary motor and fine skilled movements. Aside from that, it is also very important for equilibrium and muscle tone. So balance is also a function of the cerebellum. So the cerebellar cortex again calculates the best way to coordinate the force, direction, and extent of muscle contraction to prevent overshoot, maintain posture, and ensure smooth coordinated movement in execution of a skill. So before we proceed, what is muscle tone? So I hope that you are not confusing muscle tone with muscle strength because those are very different. Those two are very different from each other. So muscle tone is the ability of a muscle to resist passive motion. So when you're moving, when someone else is moving your elbow towards extension, that is passive movement, our muscle has this unique ability to still resist that passive motion so as not to cause damage to the muscle. Again, this is passive, so it means there's no contraction of the muscle. It's just it's the tone. There's a resistance of our muscle to passive motion. So, when we say muscle strength, that is very different because muscle strength requires contraction of the muscle and it requires force, and that's not being done with the muscle tone. So I hope that muscle tone is clear with you guys and you are not confused with muscle tone, muscle flexibility, that's also different, and muscle strength. So the cerebellum, it consists of the cerebellar cortex, just like your cerebral cortex, and it also has a white matter. So inside the white matter are four deep cerebellar nuclei, and these are named vestigial, globose, emboliform, and the dentate. Before we proceed, here's a picture of a cerebellum. So you can see here the arbor vitae, which resembles a tree branch, The white part there is called the arbor vitae. We have the deep cerebellar nuclei, which are embedded within the white matter. The cerebellar cortex, which is made up of gray matter. And then we have the different lobes and fissures. We have the anterior lobe, the posterior lobe, which is the largest part of the cerebellum. Then we have the primary fissure dividing the anterior and the posterior lobe, and the horizontal fissure dividing the superior and inferior surfaces of the cerebellum. And then lastly, we have the vermis, which connects the two cerebellar hemisphere. It's found in the middle. Here's another picture of cerebellum. Again, here's the vermis, the one in the middle. It connects the two cerebellar hemisphere. We have the lobe, anterior lobe, the primary fissure, the posterior lobe, which is more appreciated in the inferior view. It's very large. And then we have the flocculonodular lobe, a very small lobe, but very important lobe in the cerebellum that is needed for coordination, especially with the head and eye coordination. So let's talk about the cerebellar cortex. So again, this is the gray matter of the cerebellum. In the cortex, there's different layers, and we'll discuss that one by one. We have the molecular layer, the Purkinje cell layer, and the granule cell layer. In the molecular layer, it contains two types of neurons, the outer stellate cell and the inner basket cell. So please do not confuse the neurons that are found within the cortex to the neurons or the nuclei that are found within the white matter. Those are two different structures. We're talking about the cortex right now. So these are the neurons found within the cortex. So at the molecular layer, again, we have the outer stellate cell and the inner basket cell. At the granular layer, we have the Golgi cells. So here is a picture of the layers in the cortex. Next, we have the Purkinje cell layer. So from the name itself, it contains the Purkinje cells. Purkinje cells is very important cell in the cerebellum because it's the center of a efferent output from the cerebellum going towards other structures of the brain. So this is the functional unit of the cerebellar cortex because it's the only cell that projects outside the cerebellum. Without this, the cerebellum cannot send its information to appropriate centers in our body. Next, let's proceed with the white matter. So inside or embedded within the white matter are deep cerebellar nuclei. Again, this is different from the neurons that are found within the cortex. So the deep cerebellar nuclei project out of the cerebellum and thus represent the major efferent pathway from the cerebellum. The first neurons that we have discussed, like the stellate and the basket, they are all afferent, so they will receive input. But the deep cerebellar nuclei, these are efferent, meaning they're motor in function, so they will send input to other areas outside the cerebellum. But they also receive excitatory inputs from site outside the cerebellum, but again, they represent majority of the efferent pathway of the cerebellum. First, let's discuss the dentate nucleus. So this is the largest of the cerebellar nuclei. It has the shape of a crumpled bag with the opening facing medially. The emboliform nucleus, this is ovoid and is situated medial to the dentate nucleus, partially covering its hilus. And then last two, we have globose and the fastigial nucleus. Now, the important thing that you have to remember in this is the location of the deep cerebellar nuclei. So starting from the medial to lateral, we have the fastigial nucleus, the globus nucleus, emboliform, and the dentate nucleus. The white matter of the cerebellum, again, this is where the deep cerebellar nuclei, which are the efferent pathway of the cerebellum, are embedded. and it is a small amount of white matter in the vermis, and it's called the arbor vitae. That is resembling a tree branch. Here's another picture of the arbor vitae, the white part there that resembles a tree branch. That is your arbor vitae. So inside that would be the deep cerebellar nuclei. Here's another picture of a real brain, real cerebellum. Now we're done with the neurons. Let's proceed with the fibers. So we have three groups of fibers in the cerebellum: the intrinsic, afferent, and the efferent fibers. So from the name itself, intrinsic meaning it will only project signals inside or within the cerebellum. Afferent, which means it will receive information, and efferent, which means it will send information. So we have different types of fibers, and we'll discuss this one by one. First is the intrinsic fibers. They do not leave the cerebellum but connect different regions of the cerebellum from each other. Some interconnect folia of the cerebellar cortex and vermis on the same side. Other connects the two cerebellar hemisphere. So it's like an interhemispheric fiber. Afferent fiber, on the other hand, they will enter the cerebellum mainly through the inferior and middle cerebellar peduncles. Again, we have three cerebellar peduncles: the superior, middle, and the inferior cerebellar peduncles. Specifically, in the inferior and middle cerebellar peduncles are axons of afferent input. So they will send information to the cerebellum, or they will enter. These axons are entering the cerebellum. But for the efferent fibers, they will exit the cerebellum, and these are coming from the axons of the Purkinje cell. Remember, I've mentioned the Purkinje cell is the functional unit of the cerebellum because it's a major outflow cell of the cerebellum. So its axon will be the one exiting the cerebellum, and it's through the superior cerebellar peduncle that it will exit the cerebellum. So here are the cerebellar peduncles. Again, the fibers that will exit the superior cerebellar peduncle will be axons of the Purkinje cell, and those fibers that will enter the the middle and the inferior cerebellar peduncles will be sending input to the cerebellum. Next is the cerebellar cortical mechanism. We have two main lines of input to the cortex: the climbing fibers and the mossy fibers. They are excitatory to the Purkinje cells. So once they receive input, these fibers, they will send excitatory stimulus or input to the Purkinje cell. And the Purkinje cell is inhibitory. So the climbing fibers, these are the terminal fibers of the olivocerebellar tracts. They ascend through the layers of the cortex and makes a large number of synaptic contacts with the dendrites of a Purkinje cell. That's why it can stimulate the Purkinje cell. The mossy fiber, these are terminal fibers of other cerebellar afferent tracts, multiple branches, and exert a much more diffuse excitatory effect to the Purkinje cell. A single mossy fiber may stimulate thousands of Purkinje cells through the granule cells. So what it wants to say is that both of these will stimulate and excite the Purkinje cell, but the stronger stimulus will be coming from the mossy fiber. So here's a summary of the efferent cerebellar pathways. We have the globus emboliform rural, So these are the intrinsic fibers. You have the dentatothalamic, fastigial vestibular, and the fastigial reticular. So these are fibers coming from the nuclei in the cerebellum. Here's a afferent cerebellar pathway. I won't be reading them one by one, but you can read them in your own. We have the function and the pathway, the names of the pathway for each. Then we have the divisions of the cerebellum, or the lobes of the cerebellum. We have 3 lobes: your anterior, posterior, and the flocculonodular lobe. So what I want you to remember in this slide are the function of each lobe, because they differ. The flocculonodular lobe is the one responsible for equilibrium and connects with the vestibular system. The anterior lobe is the one involved for propulsive movements, such as walking, or stereotypical movements. So these are alternating, reciprocating movements. The posterior lobe, and the largest lobe, is the one that is responsible for coordination. It's the main function of the cerebellum. And it makes sense because, again, it's the largest part of the cerebellum. So it is the primary part of the cerebellum that is responsible for fine-tuning and producing smooth, coordinated movement. And again, the cerebellar hemispheres are divided by the vermis, which is found at the middle. Here's another picture of the different lobes. Please take note of the small flocculonodular lobe that is underneath the posterior lobe. Again, this is very important for the vestibular system, the balance, and even hand-eye coordination. So the posterior lobe of the cerebellum, this one, using the complete anatomy, it's very large. This is also known as the caudal lobe of the cerebellum. It receives input exclusively from the cerebral cortex, and this lobe is involved in, again, planning movement, stereotype movement that is about to occur, evaluating sensory information for action, as well as fine motor coordination. So majority of this lobe will is responsible for coordination. Next is the anterior lobe, also known as the cranial lobe of the cerebellum, or the rostral lobe of the cerebellum. It functions mainly in the fine-tuning of body movement. So this one is the specific lobe in the cerebellum that is for automatic movements, reciprocal movements, anything that needs fine-tuning of body movements, such as the trunk as well, maintaining posture. That is the anterior lobe. So it receives proprioception information from the dorsal columns of the spinal cord, trigeminal nerve, and as well as the visual and auditory systems. And that's how it can have a function when it comes to posture, because it receives proprioception information from our body so that it can respond correctly to what the body needs depending on the position. And lastly, we have the flocculonodular lobe. This small lobe is located at the posterior margin of the middle cerebellar peduncle. Very small, if you can see at the picture. It helps control balance because it has a good connection with the vestibular system, and also eye movement, specifically eye coordination. So if you have a lesion at the flocculonodular lobe, you will have what we call nystagmus. And I hope this isn't the first time you've heard that word. So nystagmus is the jerking of the eye, shaking, oscillating of the eyes, depending if it's horizontal or vertical. But either way, it could suggest that there's a lesion to the cerebellum, specifically the flocculonodular lobe, which is supposedly the one responsible for ensuring that there's good eye coordination or eye movement. So if it's damaged, there would be incoordination of the eye or the nystagmus. And lastly, here are the signs of lesions of human motor system depending on the tract or the systems affected. That's why we have to be familiar what are the tracts and the structures that comprise each motor system. So as you can see here, we have a separate table for the cerebellar systems because I want you to remember that with cerebellum affectation, it will always be ipsilateral, okay? Because the tracts coming from the cerebellum will not decussate. It did not decussate. I did not mention any decussation of fibers coming from cerebellum. So it means that if the right lobe of the cerebellum is damaged, it will present its lesion to the right side of the body as well. Again, cerebellar lesion will produce a damage to the ipsilateral side of the body. Okay, that is the main difference of having a lesion in the cerebellum. Aside from that, it's also going to present with hypotonia because, as I've mentioned, one function of your cerebellum is for improving or producing muscle tone. So if there's no cerebellum, there would be hypotonia. Okay, and then the basal ganglia, the majority of the presentation, if you have a lesion with the basal ganglia, will be movement disorders or dyskinesias, and we'll discuss that in your lecture and laboratory sessions. So that will be all for my pre-recorded lecture. I hope that you have learned something, and if you need further readings, I have uploaded copies of electronic books in our ILS, and I also shared some YouTube files that you can watch that could be very helpful in visualizing the different pathways in the motor system. Thank you and have a good day.